Okay, picking up where we left off from gene expression video number 1, this is kind of a little bit into eukaryotic gene expression. So before we talk about natural gene expression, it's useful to think about how what we are doing now with altering genomes and making clones and things like that is still a form of gene regulation. The three types that we do the most frequently are gene amplification, where we take a gene and make it more repetitive or more present. Gene loss, where we cut out certain genes, and transpose genes, where we take a genome and we can transpose it into another section of DNA in an organism or into a totally new organism. When we look at gene duplication and transposition and how it regulates genes, we're going to look at globin, specifically alpha globin and beta globin. This image depicts how globin, right? We talk about hemoglobin, which is what we have, has evolved over time. There's a specific gene called globin that was the ancestral gene, and then it began to duplicate. First, it duplicated and it was identical, and then we see it morph into alpha and then beta, and then we see it start to develop onto, it transposes. So first it duplicates, and then it mutates so that we get alpha and beta. Then it is transposed onto a different chromosome, and then it starts to duplicate and mutate into that like operon sequence. So there are multiple proteins within the alpha globin gene. We have alpha and beta globin to make our hemoglobin cluster, and the alpha globin comes from chromosome 16, and the beta globin comes from chromosome 11. The rearrangement of gene domains can also regulate genes. When we talk about beta lymphocytes, if we remember back to the immune system, we can produce millions of different types of antibodies that react with millions of different antigens. This happens by the rearrangement of your A and B genes. If we look at the image, it occurs in 3 steps. We have the DNA of an undifferentiated cell. Essentially, that means that it does not have a purpose yet. Once a section of DNA is deleted, we see that at the end, right? Technically, we're removing the introns and the exons. Then we see the differentiation of the DNA. Specifically, one section becomes the functional gene and another section is not used. So in this case, the V1 gene is no longer being used. Then it is transcribed and then translated into a gene. And so we see that not only have we cut out certain parts and it's been rearranged, but we get a totally different protein or polypeptide chain than when we started with our undifferentiated DNA. Another example of gene rearrangement and mimicry and gene regulation, you're welcome to look this up if you would like to. It's not super important though. So when we talk about this idea of gene regulation, we have to include the idea of epigenetics, which refers to the process that influence gene expression or function without changing the underlying DNA. So you've maybe heard that each gene can impact the expression of another gene, and this is called epigenetics. There are 2 processes, and these don't impact the DNA itself, but they basically attach accessory parts to the original DNA. Acetylation and methylation are the 2 processes that allow this to happen. Acetylation, the first section, and it's technically the acetylation of lysine, is found on the histones. It decreases the affinity of histones for DNA and other histones, therefore making DNA more accessible for transcription. So the way that this actually regulates the DNA is it makes the DNA unwind from the histones and makes it more readily read for transcription and translation. The other process called methylation, when we're talking about epigenetics, is that a methyl group can be added to the nitrogenous bases of cytosine that are followed by a guanine. Many human genes have upstream CG-rich regions. we call them CPG islands, but they're literally just repeats of C's and G's. And if a methyl group, which is like a little protein, can attach to this, it can impact the expression. Specifically, it can repress the reading of the gene. So we see that the CPG island in the bottom image has been methylated, and it's keeping the gene from being read. Another thing that could regulate our DNA in eukaryotic cells is the role of things we call transcription factors. They are typically occurring upstream. Sometimes they are much closer to the gene itself, but they are transcription factors, or they are sequences of DNA that impact the transcription and translation of a gene. So if we look way far upstream on this original DNA, we have these things called activators or enhancers, and then we've got further downstream my promoter, my TATA box, and then the actual operon, or the gene. What can happen is that these transcription factors, or the activators, actually double back and attach to the TATA box and the RNA polymerase, making a transcription complex that causes the RNA synthesis. And so these activators way far downstream can kind of double over, attach to the TATA box, RNA polymerase attaches, and then we have our transcription complex. Another role of transcription factors or activators, right, this very far upstream sequence of DNA, different cells have different activators which activate different genes. It's super variable, and there's not exactly one rule to describe what happens. In the liver, these liver cells need the protein albumin and not the protein crystallin, and these like lens cells do not need albumin, but they do need crystallin. And so if we look at the liver cell, which is on the left, we'll see that that way far upstream activator has doubled back. It's attached to the promoter, and albumin is transcribed. If we look at the lens cell, which is a different type of cell, we see that a whole different type of activators are going to double over. In the lac operon, or the genes, right? We talk about operons with prokaryotic cells mostly. The LCT gene produces the enzyme lactase, which digests lactose. So keep in mind that we talked about this with prokaryotic cells. It is totally different in eukaryotic cells. The lac operon is only present in prokaryotic cells. The lac gene is present in eukaryotic cells. In order for lactose to be created, or for it to be transcribed, the LCT gene needs the regulatory protein coded for by a totally different gene. Most humans, after weaning, cease to produce the regulatory protein, but a mutation in the gene will allow it to continue production through adulthood. This mutation causes a condition called lactose persistence. So another control is going to be external hormones. You can look through this on your own. Okay, so those are all pre-transcription translation controls. We have some post-transcription controls as well. One is alternative splicing. We know that we are separating the exons and the introns and then putting the exons together, which are the useful parts. Depending on what gene we are dealing with and what is considered an exon and an intron, that can determine what kind of mRNA I end up with. Again, we can see it here. I've got this whole sequence of exons, and then the blue are my introns. Due to alternative splicing, I can end up with 3 different proteins. Again, in humans, we can end up with 2 totally different proteins depending on how I've spliced my pre-mRNA. Another post-transcriptional control is that mRNA molecules do not remain functional indefinitely. The length of time that it can create proteins varies. Small pieces of RNA can interfere with mRNA by being complementary to a small part, and tagging it for destruction. Essentially, what can happen is that basically it starts to degrade eventually, and so my ability to make proteins is going to start to turn off. Another control post-transcription is microRNA, and this kind of goes back to this microRNA. Another post-transcription control is siRNA, and these are not super important for the AP test. Feel free to look them up if you want to, but don't focus on them too much. There are a couple other factors that can impact the expression of genes post-transcription. Chemical signals that regulate the mRNA leaving the nucleus. Nearly half of all mature mRNA never reaches the cytoplasm because there's some sort of chemical inhibitor that keeps it from leaving the nucleus. Degrading mRNA affects its lifespan. This also can include the degradation of its 5-GTP cap or the poly A tail. As it breaks down, the mRNA can not last as long. This is an example of ubiquitin, or the process of making ubiquitin, and we can see that eventually that DNA is slowly starting to degrade, and we make fewer of them. Other factors that can impact the expression of genes, the modification of protein products. Often amino acids have to be removed in order for their protein to be functional. Proteins are often modified with prosthetic groups to make them functional. And so the purpose of the mRNA ends up being done at a certain point. And that is it for control of prokaryotic and eukaryotic genes.